Okay, I was on a live stream a little while ago with Gutsick Gibbon and Steve McRae talking about an old favourite topic of mine, which is human chromosome 2 fusion. For those of you that don't know what I'm talking about, it's the hypothesis that explains why humans have one less pair of chromosomes, so we have 23 pairs, have one less pair than our great ape cousins, the chimpanzee, gorilla, and orangutan, that have 24 pairs. So obviously if we share a common ancestor with them, then there needs to be some explanation for why we are missing a chromosome pair. All right, so here's what we propose the genome of our common ancestor looked like, with 24 pairs of chromosomes. And then after our ape cousins branched off one by one of our chromosomes pairs fused together like so. And that would explain why they still have 24 pairs and we have 23 pairs. Now obviously young Earth creationists don't really like that idea, so people like this guy, which is Dr. Jeffrey Tomkins from the Institute for Creation Research. And he's put out a bunch of articles trying to refute the idea of chromosome fusion. Now Jeff Tompkins and I have quite the history. We've been going back and forth since about 2014, when I made him retract one of his published papers. So he would prefer that I go away, and he does his best to ignore any direct criticism of his sloppy work. Lucky for Dr. Tompkins, he has many parrots out there willing to spread his garbage, and one such parrot is Standing for Truth. Now Standing for Truth has put out a video which was a whopping 47 minutes long, and pretty arrogantly named the video that changed the world. And my response video here is really only responding to about 1 minute of his video, which reminds me of a great maxim about creationists in general, and that is, in practice, each point raised by the Gish Galloper takes considerably more time to refute or fact check than it did to state it in the first place. And Standing for Truth is just a parrot. So on screen, I've got a little snippet from one of Jeff Tompkins' articles. So have a listen to how Standing for Truth just, changes the odd word here and there and adds a few words elsewhere.
The rescue device used by the authors hypothesized that maybe the chimp satellite DNA was just randomly deleted in the fusion event. Well, unfortunately for them, the telomere sequence did stall to it. So much for that rescue device. Even despite the assumed deletions of DNA in the fusion event described in the study, their rescue device model cannot even remotely account for the 24 million of base pairs of DNA loss.
Right, but here's the actual clip that I'm going to respond to in this video, and it concerns something called satellite DNA, and whether we should find it at the fusion site.
Here are the facts. All known cases of mammalian chromosome fusions in living animals involve a specific category of sequences called satellite DNA, which is considered part of the junk DNA, but has recently been found to have a structural and functional role in centromeric function, and satellite DNA is found at and near all telomeres, which is why we should also find evidence at, near, or around this area if there was a fusion. Only telomere to satellite DNA or satellite to satellite DNA are found in the chromosomal fusion sites of living animals in nature, not ever telomere to telomere fusions. Yet, the hypothetical chromosome fusion, which supposedly took ape to man, was a telomere to telomere fusion that leaves no trace of this satellite DNA behind, like it does with all other known mammalian fusions.
Right, so here's the paragraph that he is paraphrasing from. A third major problem is the fusion site contains no type of sequence called satellite DNA. In chromosome fusion events that occur in nature in living mammals, a very rare event, the DNA signature always involves satellite DNA producing a DNA signature that occurs as either satellite DNA to satellite DNA or satellite DNA to telomeric DNA sequence. Thus, the alleged fusion event should contain satellite DNA, a problem the fusion site discoverers openly acknowledged in their initial 1991 paper. When telomeric DNA to telomeric DNA fusions do occur in humans, They involve tissues and cell lines associated with cancerous tumours. Now, before we get into the actual meat of the video, there's one quote from Tomkins that has always puzzled me. So it's this one here. In 1991, scientists reported finding alleged evidence of a telomere-telomere fusion event on human chromosome 2, but it was not quite what they were expecting. Thus, the alleged fusion event should contain satellite DNA, a problem the fusion site discoverers openly acknowledged in their initial 1991 paper. Dr. Jeffrey Tomkins. Now, the paper he's talking about is this one. It's called Origin of Human Chromosome 2, an Ancestral Telomere-Telomere Fusion by J.W. Ido in 1991. It's literally 5 pages long, and probably about half of that is pictures and diagrams and references. So it only takes about 5 or 10 minutes to read. And there's nothing at all in that paper that suggests that they were surprised or expecting something other than what they actually found. So I pulled a quote here, which kind of gives you the vibe of the thing. The inverted arrangement of the TTA-GGG array and the adjacent sequences, which are similar to sequences found at present-day human telomeres, is precisely that predicted for a head-to-head telomeric fusion of 2 chromosomes. So I really encourage everyone to have a read for themselves, because it looks like Tomkins is just making shit up here. All right. Anyway, should we find satellite DNA at the fusion site? Well, we probably should explain what satellite DNA is before we can answer that. Right, so you can divide the DNA in our genome into 2 fairly broad and inexact categories. So on the left here, we've got single copy, high complexity coding DNA. So this is usually where the genes are that make proteins. And on this side, we've got highly repetitive, low complexity, non-coding DNA. Right, so it's not that one. And you can further divide that repetitive group into 2 subgroups. So the one on the left, which is a single string of DNA that has copies in many different places in the genome. So here, here, here. These are usually called transposons or jumping genes. In the human genome, the most common one is called the ALU element. And there are literally hundreds of thousands of copies all across the genome. The other group is called the tandem repeats. Tandem because they are one after the other, so they tend to form big blocks. And satellite DNA is part of this group, long blocks of repetitive sequences. Right, so before we get to where we can find satellite DNA, I probably should give a quick rundown on the basic structure of a chromosome. If you're watching a video about chromosome fusion, then you probably already know that the red parts here are called telomeres. And they're the special sequences on the end of the chromosome that protect the genes, the internal genes, from being damaged. And if they work properly, they also stop the chromosome from fusing to some other chromosome. And what telomeres look like is a whole bunch of the sequence TTA-GGG repeated one after the other. The green bits here are called centromeres. And then you can probably figure out from the name that they're usually in the middle of the chromosome. And they're a really important structural component during mitosis. So centromeres are made up of tandem repeats as well, but the repeat unit in this case is a sequence called the alphoid sequence, and it's about 170 base pairs long, so a lot longer than the TTA-GGG repeat in the telomeres. I've also drawn 2 kinds of chromosomes here. The one on the left has genes on either side of the centromere, so it has 2 arms, and the centromere is roughly in the middle, so this one would be called a metacentric chromosome, while the one on the right, the centromere is really close to the tip, so you've got one long arm with all the genes on it, and then the other arm is really short and doesn't have anything particularly useful on it. This one's called an acrocentric chromosome because the centromere is near the tip. So whether you want to call this little grey block here, whether you want to call that close to the centromere or close to the telomere is really up to you because it is both. Right, so just straight out of Wiki, which has actually been lifted straight out of a textbook, here are the major satellite DNA families in the human genome. So we've just talked about the alphoid sequence. and that sequence is the major component of centromeres. We've got beta satellite, which is also found around centromeres. And then we've got these three other satellites, 1, 2, and 3. So where are they found? Right, now first of all, there is actually a distinction between satellites 1, 2, and 3 with Roman numerals, and satellites 1, 2, and 3 with just our normal Arabic numbers. And if you want to read this paper, you can get the detail, but for simplicity, we're just going to conflate the two. So satellite I equals satellite 1, satellite I equals satellite 2. All right, so where is satellite 1 found? So quote, the most abundant satellite 1 repeat, the ABABA form, is located at the pericentromeric regions of chromosomes 3, 4, 13, 14, 15, 21, and 22. All right, so peri means around, and centromeric obviously refers to the centromere. So satellite 1 is found around the centromeres. So what about satellites 2 and 3? All right, so here's a nice diagram from a paper in 1993 called Chromosomal Localization of Human Satellites 2 and 3 by a FISH method using oligonucleotides as probes. All right, now the scanner did a pretty bad job here, but you can see that the big blocks of satellite 2 are around the centromeres of chromosomes 1, 2, 10, and 16, while the big block of satellite 3 is around the centromere of chromosome 9. So satellites 2 and 3 are also found on the acrocentric chromosomes, which remember are the ones where the centromere and the telomere are really close together. So for those chromosomes, you could say that the satellite DNA is found both near the centromere and near the telomere. But when someone asks you where you should find satellite DNA, the answer is near centromeres. That's not to say that you won't find it anywhere else, but around centromeres is where you expect it to be. So let's go back to Tomkins. This is a slightly more recent quote of his. This is from a 2017 post called Debunking the Debunkers, and he states pretty explicitly here, all known fusions in extant mammals involve breaks at or near centromeres. So here he's explicitly saying that there are no telomere-telomere fusions in mammals. In 2018, I published a video with the title, Are Humans the Only One? Which showed that there were telomere-to-telomere fusions in at least 2 other mammals, that being the domestic pig and Hartmann's zebra. So I'll leave a link to that video in the description. What's interesting though is that Tomkins put out another blog post about chromosome 2 fusion only a few weeks ago, and he seems to have dropped that explicit claim that there are no telomere-telomere fusions in mammals, but still holding on to this expectation of satellite DNA at the fusion site, which is the point of this video. Right, so should we expect to find satellite DNA at the fusion site? So remember Tomkins said 2 things. Number 1, that we should expect a satellite DNA to satellite DNA or satellite DNA to telomeric DNA signature, and that's the claim that we're addressing in this video. And 2, all known fusions involve breaks at or near centromeres, and we know that's false because we do have examples of telomere to telomere fusions. Right, so let's have a look at the different types of fusions. Remember the red is the telomere, the green is the centromere, and I've put some yellow blocks around the centromeres just to signify the satellite DNA. So this first one, we are going to break each chromosome off at the centromere here. That one gone, that one gone, and then fuse them together. So in this case, because we fused at the centromeres and satellite DNA is found around the centromeres, then yes, we have this satellite DNA to satellite DNA signature around the fusion site. In this second example, we're just going to have one chromosome break at the centromere, and it's going to fuse with the telomere of another chromosome. So in this case, again, because satellite DNA is found around centromeres, then we're going to have satellite DNA to telomere DNA signature around the fusion site. But in this third case, and this is the type of fusion that Tomkins pretends doesn't exist, we've got 2 non-acrocentric or metacentric chromosomes. And in this one, we're not breaking near the centromere. We've just got 2 telomeres fusing. And what do you know? We don't expect any satellite DNA signature at the fusion site. So, to hear Tomkins' claim for a third time, however, the fusion signature was somewhat of an enigma based on the real fusions that occasionally occur in nature. All documented fusions in living animals involve a specific type of sequence called satellite DNA, located in chromosomes and found in breakages and fusions. The fusion signature on human chromosome 2 was missing this telltale satellite DNA. But as I've just shown, we should not expect satellite DNA at this fusion site because this fusion site does not involve a break at or near a centromere, where satellite DNA is typically found. It's as simple as that. And Tomkins should know this. Like, he's not an idiot. Dude's got a PhD in genetics. So I can't really blame standing for truth here. He's just parroting what he's been told. So if you want to go straight to the source, there he is. And feel free to ask him why he expects satellite DNA to be at the chromosome 2 fusion site.